So we don't really talk about consciousness in this class. Consciousness is actually quite a difficult thing to study. It's hard to describe, and honestly, we're not sure how consciousness arises. But what we do talk about in this class is sleep, which is a state of being unconscious. And we spend about a third of our lives in this unconscious state. And so what we can do is we can compare the brain between an awake state, a conscious state, and a sleep state. And this gives us some insights into both sleep and consciousness. So that's what we'll do in this lecture. This lecture is based on module 5.1 in your textbook. So we'll start our discussion of sleep by talking about biological rhythms. Biological rhythms can occur on different time scales. So we can have yearly rhythms. So this might be like hibernation patterns if you're some sort of wild animal. Monthly rhythms, so like the menstrual cycle, for instance. Daily rhythms, and that's where sleep comes into play. And hourly rhythms, like the urge to eat and go to the bathroom. Sleep follows what's called the circadian rhythm. And the circadian rhythm encompasses states of both sleep and wakefulness. And so psychologists and chronobiologists have looked at how circadian rhythms are regulated. How does the brain maintain circadian rhythms? Well, in the retinas of our eyes, along with photoreceptors, bipolar cells, and ganglion cells, there are a special class of ganglion cell that respond to light levels. And they send signals to the hypothalamus, in particular the suprachiasmatic nucleus, or SCN, of the hypothalamus. Now, I've mentioned the hypothalamus before. It's involved in hormone regulation. And so in this case, these light-sensitive retinal ganglion cells trigger the hypothalamus to release the hormone melatonin. Melatonin is released by the pineal gland. And what we see is that melatonin release increases throughout the day. And it really starts rising about around 10 p.m. and peaks at like 3 or 4 a.m. Taking melatonin supplements is actually something that many doctors prescribe for people who are having trouble falling asleep and maintaining sleep. And they do work, actually. If you take them a few hours before bed, you can supplement your biological stores of melatonin and help induce sleepiness. So there's some evidence here that the sleep-wake cycle, the circadian rhythm, is maintained by cues in our environment, so light levels. And we call this entrainment. So our biological rhythm, a biological rhythm I should say, in this case the circadian rhythm, is entrained to environmental cues. And so something chronobiologists have been interested in is, are there inbuilt, internal, endogenous mechanisms that help maintain biological rhythms, that help maintain the circadian rhythm, for instance. And so if we take away environmental cues, do we still see a normal cycle of sleep and wakefulness in people? And in the 1960s, a kind of crazy series of studies were run by Jürgen Aschoff. Aschoff is often called the founder of chronobiology, the founder of this field that studies biological rhythms. And what Aschoff did was he built an underground bunker and put people in the bunker and had them live there for 3 or 4 weeks without any external time cues. So no clocks, no watches. In this underground bunker, participants were told to live their lives as normally as they could. So to eat when they felt hungry and to sleep when they felt sleepy. And Aschoff measured different aspects of their physiology. So he measured more than just when they went to sleep and when they woke up. And again, the idea here is to see if in the absence of all external time cues, they'll still maintain something that looks like a circadian rhythm. What Aschoff found was that over the 3 or 4 weeks, participants tended to adopt a 25 to 26 hour day. So that was their daily cycle, and they maintained physiological rhythms that were consistent with this circadian rhythm. So for instance, changes in body temperature tracked their sleep-wake cycle in this slightly prolonged day. Now, why did they adopt a 25 or 26 hour cycle? It's not 100% clear. What we know about these studies is that Aschoff let participants control their light levels. And so one idea is that we all tend to like to stay up a bit later. And so by letting participants control when they turned on and off lights, they sort of stayed up later than they should have, left lights on longer than they should have, and this messed with what would otherwise have been a 24-hour sleep-wake cycle. This experiment was repeated in 1999, so just over 20 years ago now. And in this case, the researchers maintained strict control over light levels. So they didn't let participants turn on and off lights. The lights were kept very low and very dim. And they found that people in this situation tended to adopt a circadian rhythm that was almost exactly 24 hours. And that's what this bar plot shows here. Number of subjects on the y-axis, circadian period on the x-axis. And again, this is without external cues, and it doesn't matter if you're older, the dark bars, or younger, the light bars. Most people adopted about a 24-hour day. So although our 24-hour clock, our circadian rhythm, is maintained by external cues like light levels, in the absence of these cues, there's an internal mechanism that leads us to maintain a 24-hour sleep-wake cycle, more or less. So what's interesting about sleep is that sleep itself has cycles or stages. Your brain cycles through different stages as you sleep, and these stages can be measured in a sleep lab using electroencephalography or EEG. And so if you recall, EEG involves placing electrodes on your scalp that measure your brain activity. It has very poor spatial resolution, so we can't see exactly what part of your brain is active, but it has very good temporal resolution, so we can track broad changes in brain activity. And what we find is that you have very distinct sleep cycles that are defined by the frequency and amplitude of your brain waves. This is an image of somebody in a sleep lab. This may seem weird to sleep with this set up on your body, but these labs help sleep researchers diagnose sleep disorders like sleep apnea. So there are 4 stages of what we call non-REM sleep and 1 REM stage. And again, these stages are defined by how our brain waves look. So when we're awake, our brain waves have a low amplitude and high frequency. So they're very chaotic. They're bouncing all over the place. They're unpredictable. We call these beta waves. As you move into a sleep stage, your brain waves become more synchronized. And so we start to see what we call alpha waves. So the frequency of your brain waves decreases, and the amplitude starts to increase. This pattern of decreasing frequency, increasing amplitude continues as you move from stage 1 sleep to stage 2 sleep to stage 3 and stage 4 sleep. And indeed, in stage 4, your brain waves are called delta waves, and they have this very low frequency, so very few cycles per second, very high amplitude. They're very predictable. So stages 1, 2, 3, and 4 define what we call non-REM sleep. After you've cycled through these stages, you'll move into what we call REM sleep, or rapid eye movement sleep. And it's called that because your eyes are moving rapidly under your eyelids. And brainwaves in the state look like you're awake. So brainwaves during REM look like beta waves to some extent. We'll talk a little bit more about REM in the next lecture, but we tend to associate REM sleep with dreaming. So if you're woken up during REM, you're much more likely to report that you were in a dream than if you're woken up during another sleep stage. Another interesting aspect of REM is that movements below your neck are actually inhibited, so you experience what's called REM paralysis. And there's a disorder where people don't have this, and they actually act out their dreams, which is very dangerous. So as the night wears on, the amount of time you spend in each of these stages changes. So early in your sleep, 1 to 2 hours in, you spend a lot of time in the non-REM sleep stages, in particular stages 3 and 4. As the night goes on, you spend more time in REM sleep. And it's not unusual to wake up in the morning from REM. So if you often wake up from a dream, that suggests that you were probably in REM sleep. So as the night goes on, you experience less deep sleep and you spend more time in REM sleep. Now, the amount of time that you spend in non-REM versus REM, and indeed the amount of time that you spend sleeping, changes as you age. So this graph shows age in years on the x-axis, hours spent sleeping each day on the y-axis. What you can see is that really young infants, so babies, spend upwards of 16 hours of sleep each day, and half of that time is in REM sleep. And as you age, you spend less time sleeping and less time in REM sleep. And one of the ideas about the importance of REM is that it's involved in learning and memory. We'll talk about this in the next lecture. And so if that's the case, this kind of makes sense, right? When you're a baby, you are arguably learning the most. This is the most fruitful time in your life for learning and remembering. And so it would make sense that you're spending a lot of time in REM sleep. So we've all experienced periods of sleeplessness, insomnia, and it makes you just feel terrible, right? There's nothing worse than a poor night's sleep or going a night without any sleep at all. So what happens if you do not sleep? If you were unable to sleep for a long period of time, what would happen? Well, this is obviously an experiment that we can't run with humans. So we can't bring people into the lab and keep them awake for weeks and see what happens. In the 1980s, a behavioral neuroscientist at the University of Chicago examined what happens to lab rats when they are totally deprived of sleep. And the way these experiments worked is they used a design known as a yoked control. So the experimental rat and the control rat are connected or yoked together in some way. And so an experimental rat, the white rat here, is placed on a platform. Its brain waves are monitored using EEG. A control rat is placed on the other side of the platform and its brain waves are also monitored using EEG. And every time it looks like the experimental rat is about to drift off, so every time the EEG signal shows a decrease in frequency and an increase in amplitude, the platform would rotate. And what the rotation of the platform does is it wakes the rats up. It keeps them awake. In particular, it keeps the experimental rat awake. So the experimental rat is totally deprived of sleep. The control rat can sleep sometimes, so it can sleep when the platform is not rotating. Its brain waves aren't triggering the rotation of the platform. So one rat is totally deprived of sleep. The other rat is allowed to sleep sometimes. And what's kind of shocking about this study is that all of the sleep-deprived rats actually died within 30 days. So at least in laboratory rats, going completely without sleep for 30 days leads to death. And there was no obvious reason for them to die, so the researchers did autopsies, and there was no clear reason for why they died. Going without sleep also changed the rats' appearance and their behavior. So the sleep-deprived rats looked like they were sleep-deprived, like their fur started to fall out, for instance. They also weirdly lost a great deal of weight, and you can see that in this figure here. So we have weight change on the y-axis, and the experimental rats, the sleep-deprived rats, are represented by this solid line. And so their weight plummeted. Weirdly, their food intake, which is what this figure shows, increased. So they're eating more, but losing weight, and they eventually die. So it seems like sleep is necessary for life. And indeed, almost all animals sleep. There are even insects that sleep. I think many people have heard about dolphins that sleep one hemisphere of their brain at a time, right? So they don't drown. So one hemisphere is asleep, the other's awake, so that they can swim and not drown. So animals seem to need sleep. What happens when humans are sleep deprived? Well, we obviously can't run an experiment like this, but what we can do is look at periods in the year where humans lose sleep. So one of those periods is when we move from regular standard time to daylight savings time. So the so-called spring forward, where we lose an hour of sleep. Everybody hates this, right? You know, you're waking up at 9 a.m. for your classes, for work. Suddenly you have to wake up at 8 a.m. And so this is being studied because it's an example of mass sleep deprivation. Everybody, in the continent is losing an hour of sleep. And what we see is that following that loss of an hour of sleep, just an hour, there's a dramatic increase in the number of car accidents. So the Monday following the loss of that hour, car accidents go up. A week later, once people's sleep has recovered, they go back down to about the level they were at before this bit of sleep deprivation. In the fall, we gain an hour of sleep, so we can look at that condition as a contrast. And what we see is that when we gain an hour of sleep, car accidents actually fall the Monday after we gain that extra hour. And a week later, they've rebounded to about normal levels. So losing sleep affects our ability to drive a car negatively. Gaining sleep improves our ability to drive a car. And so this is a real-world example of the effect of sleep deprivation on our behavior. More generally, we know that a poor night's sleep impairs a number of cognitive functions. Working memory. So working memory is your ability to add sums in your head. Attention. Paying attention to lectures like this. It's easier after a good night's sleep. The ability to multitask, so to do two things at the same time. your ability to inhibit inappropriate responses. A lot of our psychology relates to actually stopping ourselves from saying stupid stuff and doing stupid things. And when we're sleep deprived, we're more likely to do those stupid things. So our ability to inhibit inappropriate behaviors is decreased. And finally, sleeplessness messes up our ability to assess risk. And so we're more likely to take risks after a poor night's sleep. So sleep is very important. And in the next lecture, I will give you some tips on how you can improve your sleep if you are a poor sleeper. And these are tips from that I've sort of put together from my own research on this topic because I am a very poor sleeper and I've suffered from insomnia for a long time. So we've come to the end of the lecture. Let's do the lecture quiz. Question 1. Our daily sleep-wake cycle is called a blank rhythm. The hormone blank helps regulate this cycle. So what's our daily sleep-wake cycle called? What hormone helps regulate it? What's our daily sleep-wake cycle called? It is, of course, called the circadian rhythm, and the hormone that regulates it is melatonin. What neuroimaging device would we use to measure the stages of sleep? So thinking about the different neuroimaging devices we've talked about and the one presented in this lecture, what is it called? What neuroimaging device would we use to measure the stages of sleep? We would use electroencephalography or EEG. Could you measure sleep stages using functional magnetic resonance imaging? Maybe, although MRI machines are quite loud and very expensive. So it typically costs about $400 an hour to use an MRI. And so to have somebody in there for a whole night would cost a lot of money. Compared to an awake state, what do brain waves look like when we're in stage 4 sleep? So compared to brain waves in an awake state, what do stage 4 sleep brain waves look like? So stage 4 sleep brain waves have a low frequency and high amplitude. So the key here is as we move from stages 1 to 4, our brain waves decrease in frequency and increase in amplitude. They become more predictable, slower in a way. When we move into REM, our brain waves become more chaotic. They look like an awake state. The reading for next class is module 5.1 in your text, so it's the same reading as for this lecture, and we will pick up where we left off and talk about the importance of sleep.